everyone, and welcome to another episode of the Ion Physiology podcast. I am Hunter, and we are going to be talking about a little bit of neuronal stuff, and then we're going to be moving from there into muscle.
Muscle, yes, good deal. Thank you, Hunter, for that introduction. Yes, once again, this Ion Physiology podcast. Please don't forget to like and subscribe. Ring that bell. Hit that like button, all the other things that we don't really care all that much about. But it's fun to say. So before we get started, there's a couple of announcements that we are going to go over. Let me pull those up. Thank you, Dom, for doing those announcements. Actually, that's for the town hall tonight, right? Okay, so no announcements this morning. We hope you're having a great week and a great day, and so we are going to jump right into it. We're going to talk about neuronal physiology, right? So neuronal physiology, and then, as Hunter said, muscle physiology after that. So let's talk about the cerebellum, the brainstem, and the spinal cord structures. Dom, why don't you take it away and talk about cerebellum, brainstem, and, you guessed it, spinal cord structures.
Alrighty, so looking directly here at the cerebellum, we know it as our, it's our master of movement.
Ooh,
so
I like that.
This is where
Master of movement.
all of our baseline movement control takes place.
So when you close your eyes and I do this, I know that my head is still tilted, even though my visual, it's you can still tell. Yeah. Okay. Yeah. Or is that more inner ear? And we're talking about coordination.
Yeah, it's
all the information, from the inner ear, from your eyes, from your muscles, spindles, from everything goes to the cerebellum.
Okay.
It basically cannot control, it's not the motor control of your muscles, but it takes everything and just makes it smooth, makes it work together in a better fashion.
You can move up the other thing.
Yeah. And then below our cerebellum, we have our brainstem. So this is where it serves as sort of the gateway to our spinal cord, and our brain and spinal cord speaking with each other, so to speak.
Okay.
And it helps also to govern posture and also help control some of our baseline. Baseline. Control of heart rate. All that.
The way I like to think of the brainstem is that it's our reptile brain. Mm-hmm. Everything that doesn't make us really human is controlled over
if we weren't human and we were reptiles,
yes,
this would control.
We will not have a frontal lobe.
Our bodies.
This is how I think about it.
I love that. That's the way you think about it.
I'm just saying, blood pressure, temperature. Breathing. It's.
Well, breathing. Yes. There's a fun. Did you have a fun fact? Did you know how they figured out why the brainstem, the control for neuronal breathing, is in the brainstem? Mm. Beheading people. Isn't that crazy? I didn't mean to make that sound exciting.
Awesome.
It didn't mean to make it sound awesome. But what they did is these, the old philosophers, right? So these philosophers, they were called philosophers. Really, they were scientists. They were medics.
Doctor of philosophy.
The doctor of philosophy, PhD. That's what it actually comes from, right? And so they looked at this and they found these gladiators that would oftentimes have injuries with swords or whatever else to the brainstem, and based on where on the brainstem or the back of the head, sorry to be grotesque, earmuffs, audience, right? If you're sensitive about this thing, they would essentially they would be dead, but the body would still be trying to breathe, essentially, and it was really based on where the injury would occur. And they actually saw more of this with the guillotine, right? With beheadings on the guillotine in the French Revolution, with what is that, the 1700s, the 1600s, 1700s?
1700s.
So, it's, and once again, this is not a history podcast, so definitely comment and let us know exactly when that was.
The grotesque thing as well, they used to actually go to people and say, when we behead you, can you try to blink afterwards?
No way, try to blink. Okay. So they would tell them, listen,
can you please
we know you're gonna die,
yes.
and you know you're gonna die.
Yeah.
Can you help a brother out, is what they said. Can you help a brother out, right? And help us with that. So that's interesting. I didn't know that they did that, but yeah, that's, die. Listen, we're all going to die one day. It's not going to be fun. It's not going to be. Spoiler alert. That's right. Yeah. But
advance the field of science.
That's right. So, and they used to do necropsies, right? So, like, instead of what we have now in medical schools, we have the gross anatomy, where we use bodies donated to science, and we're thankful for them. And they take the utmost care and utmost honor to give these people who have done this and treat these with respect and dignity. But they used to dig up cadavers, essentially dig up people, and this was not volunteers. Like, oh yeah, my uncle.
Franklin.
Yeah, I mean, they literally would dig them up and they would dissect them, and I think that Da Vinci even studied a lot of musculature. That's how he was able to just so accurately depict the human form. Yeah. With the was it Vitruvian Man, the one where he's standing like this in a circle. So. I don't know what we're talking about anymore, but let's bring it back. Let's bring it back. The reptile brain ran away with that one, and let's bring it back to the cerebellum, right? So the cerebellum is going to coordinate, right? It is going to cause fluid, very smooth movement, and then transition between one muscle group and the other. The balance, as Nadim said, is going to integrate everything. So, so that's the cerebellum / brainstem. A lot of the what do we call it? The autonomic nervous system functions are going to be there. Okay. Any other things about that?
Yeah, the cerebellum is actually ipsilateral. It's not contralateral. So,
what does that mean, Nadim?
This is something you should keep in mind, because this is really important. It's going to be on the exam, I promise you. So, contralateral.
He promises. You know it's coming.
Yeah.
You know it's coming.
Contralateral is that when your my right side of the brain controls my left side of the body. Okay? In the cerebellum, it's ipsilateral. So, the right side of the cerebellum is going to be controlling the right side of the body. Okay? Pay attention to this.
So what about the eyes? Are the eyes ipsilateral or contralateral?
They are contralateral.
Contralateral, that's right. So the left eye is actually, if you watch the optic neuronal
Absolutely.
tract, they'll cross over and will be controlled by the right side.
A little bit of it actually goes to the same
A
side.
little bit does.
A little bit.
What is it? The periphery? Is it? And I think there was
I think so,
Okay,
yeah.
so this is dating me, right? But there is an old movie about a hockey player who gets hit, kind of hit really hard, and he's lost peripheral vision. And then he still wants to do stuff, and he's got nothing to do. He's not famous, and he's not making any money. So he goes and he basically gets into figure skating with this one. I think it's called not Cool Runnings. No, this is not. This is what is that called? Anyway, audience, if anybody ever watches this, remind me what that's called. It's about a hockey player who goes. You want to just Google it real quick? We do have this thing called Google. We could do it. A hockey player that loses peripheral vision and starts figure skating. It's called The Cutting Edge. I didn't see it, but that's what it's called. Is that what it's called? Yep. The Cutting Edge? Yes! Thank you, brain. Coffee.
Did you see that? No, did you see that? That's him reaching a conclusion. That's what happens when you actually try to study. He will never forget this now.
Reptile brain.
I'm serious.
Reptile brain.
Just because you went and you looked inside his brain, this is how you should be studying. He will never forget it. He did that the other day to me in class.
Did I do it?
Absolutely.
What did I do?
When I asked you a question, you figure out, you tell me what.
Yes.
And now I will never forget.
Good. Yeah.
Okay.
Make sure you.
When you reach that conclusion, you will never forget it.
That's good. Good word from Nadim. All right, now a word from our sponsors. Just kidding, we don't have any. All right, next one. Upper and lower motor neurons. Upper and lower motor neurons. What do we need to know? First of all, what the hell is a motor? Oops, can't say hell. What the heck is a motor neuron? We'll have to bleep that one out.
Yeah, we can do that.
We can do it. All
I
right.
think it'll be fine.
So, moving on to the next point. What is a motor neuron, right? What's an upper and lower motor neuron? Go ahead, Caleb, tell us what an upper and lower motor neuron is.
So when we're looking at the actual neuronal control of muscles, we have to look at kind of where that signal originates and how you get all this innervation of muscle, how you get the actual muscles recruitment, like when you're talking about neurons innervating. So you have upper and lower motor neurons. So really simple, upper motor neurons are going to start more in the upper region. So we talk about starting in the motor cortex of your brain. So you have different areas of your brain called cortexes and like little areas, locations, regions in your brain.
Cortices, plural?
Cortices?
Cortices, or is it cortexes?
Cortices?
I've never heard the word
I'm not an
cortices.
English major.
It's cortical centers.
Cortical, but that's singular, right? No, that's plural. Cortical
If you say
center.
if you say cortical
The centers is the, okay, gotcha, yeah.
Okay.
Little English, you got a little sprinkle of little English in there.
So you have different areas that are referred to as cortex, whatever it is. So cortices.
We're going to go with cortices.
So we're gonna go with that. And so the motor cortex is gonna be where you're having, rather than like sensing stuff, you're sending out signals to actually do all these kinds of movements. So upper motor neurons, their cell bodies and their originations are gonna be in that motor cortex in the brainstem area. And essentially those are just the initial signal senders. So the upper motor neurons start in your upper region and are going to go down, whether it be through different tracts or whether it be, there are some upper motor neurons that actually just kind of stay in this area, but they're going to move down the tracts that you talk about when you're looking at the spine, when you're looking at going through like ganglions, all those kinds of things. And then those are going to innervate and attach onto your lower motor neurons, which are the motor neurons that actually go and attach to your muscles and innervate those muscles and cause that like fiber movement and recruit other surrounding neurons to cause different levels of recruitment, because you're going to have like finer movements. You don't need as much innervation. You want very little fibers moving for like fine movements if I'm trying to like turn a little screw or something, whereas if I'm trying to throw a punch at somebody, I probably just want like a lot.
Jump straight to the
boom.
violence. You just jump straight
Haymaker.
to
That's what in the micro.
See, this is the problem with today's young generation. We jump straight to the violence. This is crazy. I'm just kidding.
In the micro
so
I refer to
is there a haymaker? Haymaker,
Yeah.
yeah. You know what's interesting? Why is it called a haymaker? Do you guys know why?
I don't know why.
And neither do I. Okay, moving on. We should look that up. Haymaker, right? Haymaker. I mean, I think with these guys like farm boys,
Like
initially,
throwing
right?
hay.
Throwing hay does. You gotta kind of throw. But I thought haymaker's like just a really strong punch, right? We don't have to. I mean, we can look it up, but we don't have to. Okay, so let me ask you this: is there a specific name for the connection between the upper and lower, where those two neurons meet? Where we have like a neuron and a neuron, and they kind
Synapse?
of right. It's a neuronal synapse. I was thinking of the word ganglion. Right? Is that not what that refers to?
Well,
Or
that's
am I
what,
thinking of something
that's
else?
what I was talking about, is when you're jumping from your upper motor neurons, it goes through these pathways and these ganglia, goes through like the ganglion, which is just, if you're looking at the nervous system and specifically along the spinal tract, ganglia or a ganglion, they look like these little nodules, which are kind of in a chain. So we call it a chain ganglion. Chain ganglion.
Chain gang.
What a chain ganglia, a chain ganglion. And so if you're following along the spine, actually, so we were talking about gross anatomy. If you're following along the spine, it looks like little pea pods that just follow along the spine. And essentially what that is, it's a sac where those nerves actually latch onto each other, and it's like an elevator
Connection,
system.
right?
Yeah.
A little connection point, right?
Yeah.
Gotcha. Okay, good deal. And that's where you get the pre and post ganglionic
Yes.
aspect.
And
of
then
that
where it
phrase.
meets with the muscle, where the lower motor neuron, that's going to be your neuromuscular junction.
NMJ. What?
Yeah.
I get excited about the NMJ. It's very unique. I mean, it's electrical, right? When you think of like what a neuron looks like, right? It's very, it's bizarre. Like they have a very odd shape, and they can be shaped all different things, right? A normal cell, you think, oh, it's a square, or a circle, or squarish, or circle-ish, or spheric, something like that, right? Neurons are just crazy different. And so and also what do they do? They don't contract, right? Therefore electrical. And so there has to be some way we can have the electrical communicate with the mechanical, right? The electrical mechanical coupling. And it happens at the level of the NMJ, the neuromuscular junction. And it's pretty cool, right? It's pretty cool. And we can go over that a little bit more when we get into muscle. But all right, moving on. Anything else on what was that? That was the upper and lower motor neurons.
Yep.
No.
Mm-hmm.
All right, pyramidal and extrapyramidal tracts. What do we think about those?
Hunter?
was. So I mean,
You got thrown. I don't know if you knew. You just got thrown under the bus, and I love that you just picked it up. Just picked it up.
so
He explained
when,
it to me
when
earlier
we're
this
looking at these two, they're spinal tracts. They are basically a set of neurons going through your spinal cord. For example, like the pyramidal system, we have the corticospinal and the corticobulbar tracts. These are specifically going to have direct voluntary control, and they are really important for our fine motor movements. So, like finger dexterity, you want to be able to really have fine control of those. That's going to go through your pyramidal system. The extrapyramidal system, those are the ruberospinal, reticulospinal, vestibulospinal, and tectospinal tracts. And
Techno?
Tecto.
Tic-tac-toe? Like tecto?
Yeah, close enough.
Okay. Good.
So
Good to know.
these are really important for balance and posture, and then kind of just in general orienting ourselves to our environment. So like just generally like shifting, if you hear something coming towards you, like it kind of moves out of the way without you really thinking about it.
Like
Almost
that
like
innate.
a reflex?
Yeah.
Which is the next one, right?
Mm-hmm.
Yeah.
Which is the next
All
time,
right.
right?
All right. So, reflexes, basically it's when you put your hand on a stove and hot stove, and you actually, before you even feel it in your brain, you're like lifting your hand. That's called a reflex. There are two types of reflexes we're going to be going over in this course, which is the muscle spindle, which is a little bit of sensories that are in our muscles, okay? And what they do is that they sense where our muscles are in space. They teach us how to be aware of everything. They also have they can play a different role as well. So again, when you put your hand on a hot stove and before you even feel it, you remove it, that's the extrafusal muscle, okay? Telling your spinal cord actually at this moment to remove your hand from danger, and then it will send the brain the signal after that this is what just happened. And then you have like sensors in the Golgi, they call the Golgi tendon, which are actually in the tendons. They're also muscle. If you're lifting something really heavy and your muscle is feeling that it's actually going to rip off from your bone, it will send a neuron signal to your muscle as well to just relax and let go. Drop the weight before you do so much damage. That's what happens when we're, sometimes when you're trying to lift weight and you just can't, you reach a point where you just get, you're trying really hard, but it's no, and it just drops. This is the Golgi tendon.
Story time with Dr. Brainerd. We had a wood burning stove growing up, right? So we would burn wood in the wood burning stove, hence the name wood burning stove. And it would heat the house, and we moved, and Dad wanted to take that Fisher, it said Fisher on it, right? It was a Fisher brand wood burning stove, and I don't think I've lifted anything more heavy than that. It was me, him, and a couple of my brothers, and this thing, it must have weighed 700 pounds. It was solid cast iron, just one unit, and we didn't have a dolly. We didn't even have a dolly. We had to lift that from the basement up, and at that point, we were the dolly. Yeah, he's like, I have 6 boys. I don't need a dolly. Yeah, I'm one of 6 boys. Yeah, so we had some haymakers and in our early youth. So that's what I think of when I think of an example. Or if you've ever had to move a pool table, like a real pool table, those are just some of the heaviest items that I think people potentially routinely have in their household that they can potentially relate to. But
anything that's heavy that you feel you got it, you're still holding, and then all of a sudden you feel like you've dropped it. That's not really under your control. That's really a sense that your muscles
your bones have snapped. And your muscle
it's time. It's time to save the muscle. Drop the thing off. It's time to save your functioning muscle and bone connection.
Well, and thankfully we have multiple muscles that do the same thing, right? So when we talk about the quadriceps, it's actually broken up into several different, at least in the leg area, different muscles there. and we can recruit those other, not only the myofibers, right, the muscle cells within that muscle more and more as we need to lift load, but we have other muscles that will help us do that. So when we lift something heavy, we aren't just using our legs. We're using our legs, we're using our calves, we're using our chest muscles, you're using your arms, you're using your stabilizer muscle. You are straining with everything you have, right? And so cumulatively, those can have a very powerful effect. So, okay, great. All right, what is the next topic here on the list?
Next we go over autonomic transmission.
All right, autonomic transmission, SNS and PNS. Somebody review that real quick.
Yeah, I'll go. All right, so looking at autonomic transmission, we're talking about sympathetic, parasympathetic. So if we go back and review a little bit, somatic, which is our voluntary, will have a neuron that is heavily myelinated, and it's going to be going from our CNS directly to the neuromuscular junction to have an effect on skeletal muscle, which we'll get into a little bit later. But so we have heavily myelinated. We're looking at our autonomic. So these are going to be lightly myelinated, at least for our preganglionic fibers. So looking specifically at our sympathetic nervous system, these will have short preganglionic fibers that will originate in our central nervous system, and they will synapse in these chain ganglia, and they will synapse onto post-ganglionic fibers. This is via a nicotinic receptor, so these are all cholinergic. So the preganglionic fiber is short, lightly myelinated. It will synapse on the postganglionic fiber, which is unmyelinated and will go and have an adrenergic effect on our nervous system. There's a second kind with our sympathetic, which also is preganglionic, that is lightly myelinated, and instead of synapsing on a postganglionic fiber, it will go directly to our adrenal medulla and have an adrenergic, or it releases acetylcholine to have an adrenergic effect there. Whereas looking at our parasympathetic system, these have long preganglionic fibers. So sympathetic, they synapse in these chain ganglia first, and they don't go directly to the organ they innervate. Parasympathetic goes to the organ they innervate. So they have these long, lightly myelinated preganglionic fibers, goes to the ganglia that's in the organ they innervate, synapse on the short, unmyelinated postganglionic fibers, which is all cholinergic and parasympathetic to have their effect.
Go study the material. You'll understand what
Yeah,
he's saying.
I was going to say, just trying to,
It's word vomit.
Good study. Good
You did it perfect. Seriously.
It was fantastic.
Yeah.
It made my day. I mean, it was really good. Covered all the bases, so very good. Brief review, right? So now we're going to shift gears a little bit and move over to muscle, right? So once again, this podcast, we're not reteaching the material. We're kind of hitting the highlights and going over it. So while you're brushing your teeth, you can listen to this, you can watch it, hopefully be a little bit entertained to some degree, and then but also get a little bit of physiology, right?
Listen to it to go to sleep.
Go to sleep.
Repeats in your brain.
That's right. That's right. Just hit the repeat. Just make sure the little one comes up. Isn't that the repeat? Mm-hmm. Just the same track. Right?
Yeah.
It'd be funny if you just forgot to do that and it would just went off onto like all these different podcasts. You're like, I had the weirdest dreams waking up the next morning. Okay, so let's move on to muscle. So there are three main types of muscle. They are skeletal, smooth, and cardiac. Very good. So when we just look at these muscle types, right, we can tell the difference between them, at least one of them differentiates itself, in that there is no striations. And you guessed it, it's called smooth, right? So smooth does not have the striations. And those striations, which are present in the cardiac or the cardiomyocytes and the skeletal muscle, are due to what we call sarcomeres, right? So sarcomeres are the functional unit of the muscle. Sarcomeres are the functional unit of the muscle. The functional unit of the muscle are called sarcomeres for the cardiac and the skeletal, right? Smooth muscle does not have sarcomeres, right? So let's talk about skeletal. Let's talk about skeletal first, because and when we review muscle physiology, 60% ish of the lecture is going to be over skeletal. About 30% is going to be over smooth, and about 10% or so is going to be cardiac. Simply because we cover cardiac. Cardiac has its own sort of discipline, its sub-discipline in physiology. So let's talk about skeletal. So we were just talking about neuronal, right? And now we're talking about skeletal. So before we get into the connection between those two, which is what we kind of previewed before, the neuromuscular junction, let's talk about skeletal. Like, just tell me, y'all, what are some distinguishing features of skeletal muscle?
I mean,
go ahead, Hunter.
To me, like, one of the key things with skeletal muscle is that you actually have voluntary control over it.
There we go.
You can choose whether or not a skeletal muscle is going to move.
I can choose to pump my heart right now.
Yeah. Well, I don't know about that one.
Oh yeah, I don't know. It's a superpower. Unless you see your boo, right? When you see your boo, then your heart might cardio accelerate a little bit, right? Or you can think about your boo.
A little bit of tachycardia.
A little bit tacky, right? Yeah. So you don't really have direct voluntary control, but yeah, like you said, smooth or skeletal muscle, you're gonna have voluntary control. So what are some other distinguishing features about skeletal muscle? Because remember, our audience, they should have already gone over, or at least be going through muscle as they watch
You don't need
this.
outside calcium.
You
Most
don't
of their calcium
need
comes-
outside calcium. So when we talk about muscle, the linker molecule, right? There has to be some sort of linker molecule between electrical, okay, and mechanical. So we have electrical cells. They undergo action potentials, right? Action potentials, and that action potential is enough to move the signal from one cell to the next cell, right? But what is so interesting about muscle, and especially skeletal muscle, let's kind of zoom in there a little bit. We need a molecule that is able to be released with the electrical, the action potential, but will affect the mechanical, will cause mechanical contraction, and that molecule is calcium. That's sort of the translation molecule. If you think of neurons speaking a language, and then you think of muscle speaking a different language, the translation molecule is going to be calcium. So we can jump into the neuromuscular junction if we want to. So we've got this junction between the cells, and it's important to know that this junction cannot be a physical contact, right? Anytime you have a physical contact and you have something else physically connected to that one, if this one gets shocked, it's going to transmit no matter what. You don't have a choice. Don't tase me, bro, but if you did and you're holding on to somebody else, both y'all would get shocked, okay? Both would get shocked because the electrical signal would transmit from one body into the next because of that physical contact. So it's important that we have a system that's really, really close, but it's not touching. And that's where the neuromuscular junction comes in hand, right? So the neuromuscular junction, essentially, you've got a neuron, and it's really, really close, right? It's really, really close to, that is the axon, what we call the terminal bouton, is really close to a muscle cell, right? It looks like it's almost touching it, but there is a gap of about 20 to 30 nanometers. Translation, it's okay if you don't know what a nanometer is, it's really small, right? But there's a gap there, there's a space. And so what happens is, as that signal travels down the neuron, right? Say this is the muscle right here. It travels down the neuron, it goes on the axon, and then it goes to the terminal bouton. Then, this is really cool, and I think we talked about this on the last one as well. You've got, so I'm not going to go into detail on it, you've got acetylcholine essentially is released into the synaptic cleft, right? It's released there, and then it migrates via diffusion. It's not pumped across, it doesn't fly across, it doesn't get shot across like a cannon. It's simple diffusion. It diffuses across, binds to non-specific cation channels on the motor end plate, right? That's what it's called, the end plate, and which is a special area of the muscle, and that is going to cause muscle contraction because that's going to open up where it's going to allow these non-specific cation channels to open up, allow sodium to rush into the cell, start an action potential, right? That's a neuromuscular junction. Now, here's the thing. Let's shift gears back to skeletal muscle. Skeletal muscle has a really unique transmission pathway, which are deep invaginations into the actual muscle itself, called what? What is it called? The T-tubule system. Think of it as like if you make bread, right? I love to make bread, right? If you have bread and you got the dough and you take your fingers and you're just like, right? You just stick your fingers in the dough, right? And then you take it back out. You've made a really deep indentations. Think of like the T-tubule system is a way for the action potential that is translating or transmitting across the sarcolemma to delve deep into the muscle so that you get a more consistent, more unified contraction of the entire muscle as a whole. Now, we just said we need calcium. Calcium's the linker molecule, right? If there's an electrical stimulus and we want mechanical, we have to have calcium. So what happens with skeletal muscle is skeletal muscle has in the T-tubule system, remember we're still outside of the muscle cell, it has what are called dihydropyridine channels. Now these dihydropyridine channels have many different names. They have 4 main names. They can be called a voltage-gated calcium channel, right? They can also be called a CAV 1.1 channel, and they can also be called L-type, stands for latent or slow, L-type calcium channels, right? They're not really slow. It just means they're slower than the fast voltage-gated sodium channels, so they call them L-type calcium channels, right? So you have these dihydropyridine channels on the sarcolemma, specifically in the T-tubule, right? Now, these dihydropyridine channels are in skeletal muscle, mechanically linked to other channels on the SR, right? Called what? Ryanodine channels or ryanodine receptors, right? And so what happens is the dihydropyridine channels in skeletal muscle, here's the important part, please listen up. In skeletal muscle, the dihydropyridine channels in the T-tubules act as voltage sensors. Okay? Once again, the dihydropyridine channels in the T-tubules in skeletal muscle act as voltage sensors. Okay? Now, what does that mean? It means that they sense a voltage change being transmitted through across the sarcolemma down the T-tubules, and they're going to undergo a conformational change, but because they're mechanically linked, right, to the ryanodine receptor, it's going to cause the ryanodine receptor to open up, and then calcium from the SR will flood out of the SR, down its concentration gradient, into the inside of the cell, okay? So that means that the majority of calcium coming from a skeletal muscle contraction is coming from the SR. In fact, we know this because you can take skeletal muscle, put it in a solution of darn near 0 calcium, and it can still have the ability to contract, okay? All right, so we've got that as the mechanism for skeletal muscle contraction in terms of where does the calcium come from, right? And then, have we gone over the actual actin myosin contraction? Did we go over that last time? It's on here. Great. Okay.
Talking about it today.
All right. So now we have calcium rising inside the cell, right? Is that the same mechanism for cardiomyocytes?
No.
Are you talking about with like ECC and cardio?
Right. So if we talk about where the calcium comes from in cardiomyocytes, we just said in skeletal it comes from the SR, but what about in cardiomyocytes? Somebody tell me where the calcium comes from in cardiomyocytes.
Calcium-induced calcium release.
Okay, what does that mean?
A little bit comes from outside, and it will actually lead to more coming from the SR.
Okay,
So
what's the difference between skeletal, the setup that we just described in skeletal, with cardiac?
Ryanodine receptors on skeletal muscles do not let calcium from outside the cell. In the heart, they do. Remember that.
The dihydropyridine?
Dihydropyridine,
yeah.
Dihydropyridine receptors in the skeletal muscles do not allow calcium to come through them to inside the cell.
Now, a little caveat. Could some molecules of calcium go through there? Sure, right? It's hard to say absolutes in physiology. But to Nadim's point, by and large, the vast majority, almost all of the calcium in a skeletal muscle depolarization, right, or a skeletal muscle contraction, all of the calcium comes from the SR for all intents and purposes. Okay, so in cardiac, though, you just said the dihydropyridine receptor in the T-tubule, right, actually acts like a voltage-gated calcium channel, right? So it opens up, once it senses a voltage change, calcium from the outside of the cell moves down its concentration and electrical gradient into the cell, and that is going to, now you said it was like only a little bit. What do we call that little bit of calcium that comes in? What's the term we say for it? What do we call it?
Trigger calcium.
Trigger calcium, very good. The trigger.
I was like, oh.
Yeah, oh, trigger. Yeah, so it's a trigger calcium, right? So that's the trigger calcium that's going to trigger essentially, as Nadim said, the rest of the calcium to be released from the SR, because that calcium from extracellular, the trigger calcium, will bind to the ryanodine receptors. It will cause those to open up, and even more calcium flows from the SR. So what percentage do we usually say comes from, like, what percentage of the total calcium is trigger calcium? What percentage is not trigger calcium?
Around 10% somewhere. I mean,
The important part is that it's a little bit, but it is necessary. You must have that trigger calcium in order to undergo a cardiomyocyte contraction, right? So it's a little bit of calcium. Some texts will say as little as 5%. Some will say as many as 20%, right?
Yeah.
So, but at the end of the day, it doesn't matter what it is. You must have that trigger calcium to start the cascade or start the ball rolling down the hill. Okay, so we've got that trigger calcium coming in. Okay, so that's a cardiomyocyte. Now, one of the other differences between skeletal muscle and cardiomyocyte is something we call like a triad versus a dyad, right? A triad is in skeletal muscle, but the dyad is in cardiac, right? Okay, so we've talked about that. We have calcium flooding into the cell, right? Let's talk about the mechanisms by which calcium floods into the cell for a smooth muscle. What is it, Hunter? That was a great hiding that yawn, too. That was fantastic. I mean, it was... That was fantastic.
I was doing
Kudos to you. All right, yeah, Hunter. Or we pass it on to Caleb. Caleb seems ready. Caleb, what is the?
Caleb was ready on that one.
Oh, Hunter was ready on that one.
No, I think Caleb is.
No, Caleb's ready. Caleb, tell me the 3 ways with which with smooth muscle has or allows for calcium to enter. Tell me one of them, just one of them. Ways that calcium enters the cell.
I mean, there's the same kind of like you have ligand or hormonal channels that allow calcium to go in. But then we also have the main one that we always talk about with smooth muscle is IP3 or IP2. Sorry,
IP3. You got it right.
IP3.
And also
I was counting the 3.
I saw you do too,
right,
and I was like, maybe I don't know what I'm talking about.
IP3. So let's talk about the IP3 mediated pathway. So the IP3 mediated pathway essentially is a ligand-gated pathway. You have a neurotransmitter, a hormone, a neurohormone, something like that, right? It's going to bind as a ligand, and that's going to be associated with the G protein coupled receptor. And we can get into the whole, and you do need to know how that mechanism works, but the end result is the creation of IP3. How did IP3 get there? Well, you've got this other molecule called PIP2 or PIP2. PIP2 is cleaved, into DAG, diacylglycerol, DAG, and then IP3, inositol triphosphate. This IP3 acts as a second messenger, right? What are the 2 things that a second messenger does? It amplifies the signal, meaning it makes it bigger, right? And it speeds up the transmission, right? So it amplifies and speeds up the signal. So those are the 2 main things. They can do other things, but when I was learning this back in the day when you had to walk uphill both ways, barefooted in the snow to school, that's what we learned. 2 things, right? It amplifies and it speeds up the speed, right? The velocity of the signal transmission. So we have IP3. IP3 will then go to the SR and bind to ryanodine, and it will cause calcium to be released from the SR, right? So that's a mechanism by which we can have intra-SR calcium, right, be released and cause intracellular calcium to increase. That is 1 out of 3 mechanisms. What's another mechanism for smooth muscle calcium increase?
We've got ligand-gated, like Caleb mentioned, and voltage-gated.
Voltage-gated. So what voltage-gated is what we all know what voltage-gated is, right?
Well, we all know and love.
That's right. We all know and love it, right? Good old voltage-gated, right? So the voltage travels through as an extra potential on the smooth muscle cell. The smooth muscle senses it, right? And when we say senses it, we mean the channels, the voltage-gated calcium channel, similar to the dihydropyridine channel. It's going to sense it. It's going to open up. Calcium's higher on the outside, both in concentration, and then the inside of the cell usually is negative. Calcium has a positive too, so it's going to move down its electrical and its chemical gradient, flood into the cell, increase extracellular calcium. What's the last way? So we have the IP3 mediated, we have the voltage gated. What's the last way? There's another way.
I mean, the hormonal ligand.
Yeah, just a straight up ligand channel. Okay, so it's a ligand channel where you can have a hormone or a neurotransmitter or some other metabolite acts as a ligand. The ligand binds to that channel, okay, just like a lock and key. And then what happens? That channel opens up and you have extracellular calcium flowing in. The distinction between the ligand gated with IP3 and the ligand gated just channel is where the calcium comes from, right? So the ligand gated channel, it's just going to open up. But the ligand gated with IP3, it's going to bind a ligand, right? Or a ligand will bind, but that will cause a series of events that releases or increases, not releases, but increases the amount of IP3, and that IP3 will bind to the sensors it'll bind to the ryanodine receptors on the SR, and it will release calcium causing intracellular calcium rise. So, smooth muscle, 3 ways in which we can increase calcium, right? Okay, so now let's talk about what's next. In all the muscle cells, regardless of the way, regardless of what it is, we have calcium that's increased inside the cell. What happens next? Let's talk about skeletal muscle first. Go ahead.
Actin, myosin,
So before we get to actin and myosin,
Oh, you have to troponin.
right?
You
No,
wanna
no,
troponin.
no, that's fine. You were going on the right track, right? So
Yeah.
we have calcium binds to what?
Troponin C.
Troponin. So troponin, first of all, is what we call how do we describe that molecule? It's a what?
Trimer.
Trimeric.
How many units does it have?
3.
It's a trimeric, right? So it's we can often refer to it as a heterotrimeric, right? Hetero meaning different parts, homo meaning the same. So we've got a homotetramer, right? In the ryanodine receptor, that's what it's made up of. It's made up of like 4 podocytes, 4 extensions, and it's a homotetramer, right? So it's all of those are the same, but it's 4 of them. So homo same, tetro, 4-mer unit, right? So, but in this instance, we've got troponin in skeletal muscle, and it is a heterotrimeric protein, right? So we've got 3, it's hetero meaning different, and then tri meaning 3, so 3 subunits. What were the subunits again? It's skeletal muscle. C, which is going to bind calcium. T, which is going to bind, or be bound to,
Tropomyosin.
is it tropomyosin or is it troponin? They both start with T's.
Tropomyosin.
Tropomyosin, very good. And then we've got I,
Which
inhibitory.
It covers up the binding site on the
And
tropomyosin.
exactly. So it's going to cover up and act as an inhibitory fashion, right? So skeletal muscle, in order for the troponin to be activated, you need how many molecules of calcium?
4.
You need 4 molecules of calcium to bind.
But only 3 in cardiac.
But only 3 in cardiac, right? And this makes sense. This just makes sense because it's more important that your heart beat in low calcium or have the ability to beat and contract rather than you being able to twitch your big toe, right? Your skeletal muscle, right? So it's good that there is a lower threshold for contraction to occur in cardiomyocytes as opposed to that. Okay, so calcium binds to troponin. Troponin shifts tropomyosin out of the way. Tropomyosin is rope-like protein that is blocking the binding sites on actin, okay? The binding sites that myosin needs to bind to in order to have contraction occur. So calcium binds to troponin, troponin shifts tropomyosin. Once tropomyosin is out of the way, now the myosin head and can interact with the active sites on actin and cause ratcheting. It can undergo what's called the contraction cycle right here, and it will ratchet, shortening the sarcomere as a whole.
Cross bridge.
Yes, sorry, the cross bridge cycle. What is it? Contraction cycle? Yeah. So the cross bridge cycle. Thank you for that. And so it's going to shorten the overall sarcomere, right? This is very similar to what happens in skeletal. This is exactly what happens in cardiac, and also is what is going to happen in smooth muscle, right? It still has this cross bridge cycling. Now, smooth muscles are going to have a different. They're going to have a different sort of orientation. They don't have the sarcomeric structure. They still have actin and myosin. Okay, they don't have troponin. What do they have?
Calmodulin.
They have calmodulin. Very good. So it's a regulatory protein, but it's different, right? So let's walk through how you get activation of that regulatory protein in a smooth muscle. We just talked about it, what it is in a skeletal muscle, which is the same thing in cardiac, right? What about smooth? Take it away, Caleb.
Well, so with calmodulin, essentially just calcium's floating and it binds to calmodulin, which is also just floating around.
Sure.
And then what calmodulin is going to do is that is going to wrap around a different protein called MLCK.
Which stands for?
Myosin light chain kinase.
Kinase, pause. What is a kinase, guys? You guys know this, right?
Adds phosphate.
A kinase is an enzyme that its sole duty, right, is to add a phosphate to something, right, to whatever its target protein is. So a kinase adds a phosphate, and what removes it? Phosphatase, right? So a kinase adds it, a phosphatase removes it. Okay, so Caleb, we've got calcium binding to calmodulin. Calmodulin, you said, is going to wrap around or activate
MLCK.
myosin light chain kinase.
And then myosin light chain kinase is going to add a phosphate, not just to like any area of myosin, but specifically the regulatory light chain. And so that is going to be what causes that step of cycling in smooth muscle, is because without that phosphate group, you would not be able to have the constant cycle flowing in smooth muscle with the contraction.
Very good. So you get phosphorylation of the regulatory light chain on the myosin molecule. That's going to allow for the cross-bridge cycle to occur, which we need ATP to do that, right? Because it's an active process. It takes energy to do that, right? So we've got this in smooth muscle. We've got this slightly different mechanism, which you do need to know about compared to cardiac and compared to skeletal, right? There are some other intricacies, such as when we go through the contraction cycle, the cross-bridge cycle, make sure you know the steps of that, right? Briefly, what's happening? Let's start with, you've got your myosin head is not attached. If this is actin and this is your myosin right here, you've got your myosin head is not attached to actin. We're going to start right there. So what happens next? You've got ATP coming to the myosin head, and it's going to bind it, right? So ATP binds to that myosin head, right? That's going to cause it to detach, essentially, from the actin, right? So now we've got, and this is, side note, little story time with Dr. B, this is why rigor mortis occurs, right? Because you don't have ATP, that myosin head is bound to the actin, and it can't release, because binding of ATP to that myosin is what causes that release, okay? So we're going to start right there. ATP binds, it releases from there. What happens next?
The ATP is hydrolyzed.
It's hydrolyzed. What does that mean, hydrolyzed?
So it
What that word mean, Hunter?
It breaks apart the ATP into an ADP and a singular inorganic phosphate.
And what does ADP stand for? Well, what is the A part? Adenosine, right? Right? So it's adenosine. So if you think of ATP, it's adenosine triphosphate. Adenosine molecule linked to one phosphate, linked to two phosphate, and then another phosphate. So that's ATP, adenosine triphosphate. So it's adenosine molecule with three phosphates in a chain linked right there. What we're going to do is we're going to break a bond on that between the last two phosphates. When you break a bond, you release energy, okay? Breaking a bond releases energy. So what happens is we capture that energy and we use it to do work in the cell. So that's why ATP is kind of like the fuel of the cell, right? So ATP binds to the myosin head, it detaches from actin, okay? ATP, as Hunter so eloquently put it, is hydrolyzed. That means that we're breaking a bond, releasing energy. So now we have ADP, that is in D as in dog, adenosine diphosphate, and we're using that energy to what?
It cocks the head.
Cocks the head, right? We're ratcheting that head back. Think of this as like, this is a rubber band that has already not been stretched. Right now, we're using ATP to stretch that rubber band or stretch that spring, right? So there's a lot more potential energy in this myosin head now that it has been cocked, if you will, by the hydrolysis of ATP into ADP and inorganic phosphate. So now we have the cocking of the head. Now what happens?
It attaches again.
So once again, there is a weak connection or a weak attraction for this myosin head. Now that it is cocked, right, it's going to bind weakly back to actin. Not anywhere on actin. Where on actin?
The
active sites that were exposed by calcium binding to troponin and troponin shifting tropomyosin in skeletal and cardiac. Or you could say that there's not really, similar aspect in smooth muscle, but it's a little bit different, right? Let's stick with skeletal muscle and cardiac here. So we have a weak cross-bridge formation forming, right? Then what happens?
The inorganic phosphate.
The inorganic leaves, poof, goes off. and creates a strong formation, right? And then one of the last phases of the cycle is that as ADP leaves, right, that's going to result in the what we call the power stroke, the actual movement. And you see, look at my elbows. See how long that is? If I move this over this way and do a power stroke, my elbows are now closer together, right? And so what happens is it does this, and then it just does this. It goes through another contraction cycle and does this. And so the elbow length was the length of the sarcomere, and we have shortening of the sarcomere when calcium is there. Okay. All right, so we've talked about that. We've talked about the regulation. How do we get rid of the calcium?
I mean, you have like...
Because, right, we've depolarized the cell, right? And started, I was going to elaborate on this. We've depolarized the cell. Set the stage for your response here, no pressure. We've depolarized the cell. Calcium's jacked up. We've got contraction, and now we want to relax the cell. Well, how do we do that? Well, we get rid of the calcium. So how do we get rid of the calcium?
I mean, there's a lot of different ways.
There's a lot of different ways. Give me one, Caleb.
The one we always talk about is our Circa pump, which is bound. So it's a pump on the actual membrane of your sarcoplasmic reticulum.
Yep.
And that's just the main pathway that calcium is being pumped into the sarcoplasmic reticulum.
From where?
From the cell.
From the cytoplasm,
Yeah.
so from the inside of the cell. And so that's how we, remember, inside of the cell levels of calcium are now really high compared to what they were. So now we need to bring them back down to low levels. So the Circa pump, that is SERCA, Circa pump, sarcoendoplasmic reticulum calcium ATPase pump. It's an ATPase, so that's an active transport. We are pumping, we're burning ATP, right, using that energy to pump calcium from the cytoplasm into the SR. Now, whenever we talk about Circa, we have to talk about its buddy, right? It's break. It's buddy break. What protein am I thinking about when I say Circa has a buddy that puts the brakes on? Phospholamban, or PLB.
I don't know how to pronounce
So
it.
phospholamban is constantly, if this is Circa, phospholamban's over here bound to it, and it's saying, whoa, dog, whoa, down, hold on, slow down there, horse, whoa. We just got a horse, so it's like, whoa, you gotta do that, right? You gotta pull back on the reins. This is Circa, phospholamban, when it's bound, it's constantly saying, slow down, right? Because it doesn't make sense to have Circa going 1,000 miles an hour when you want calcium levels to rise, right? We don't want it to be Circa pumping super fast, right? So phospholamban is going to be bound to it, slowing it down. But during what we call calcium resequestration, we want to get the calcium out of the cell. We just want to get it out of the inside of the cell. Whether it goes outside the cell or outside of the cytoplasm into the SR, we want to get rid of it, right? So we have to dissociate Circa from phospholamban, or I guess more accurately, phospholamban needs to dissociate from Circa. So what's the mechanism by which we can sort of remove the brakes?
That's what phospholamban gets phosphorylated.
Very good. Phospholamban gets phosphorylated by what? What phosphorylates? Yeah, you guys know what it is.
Phosphatase?
Is it PKA?
It's definitely not a phosphatase. Phosphatase removes a phosphate.
Is it
phospholamban kinase?
Okay, so it's a kinase, PKA, right? PKA, protein kinase A, right, is going to phosphorylate phospholamban. It's going to add a phosphate. That phosphorylation causes dissociation of phospholamban from Circa, and now we are letting the horses out in the field. We're loosening the reins, and we're saying, yeehaw, let's go, right? Just picture that. We ride our horses saying yeehaw, and we do say the yeehaw, right? So phospholamban, once phosphorylated by protein kinase A, is going to dissociate from Circa. Circa is going to be going super fast and moving calcium from the cytoplasm into the SR. What are some other mechanisms?
NCX.
So there's something called the NCX, the sodium calcium exchanger. And the sodium calcium exchanger is on the sarcolemma, or the cell membrane, the plasma membrane of the muscle cell. And it's going to pump, essentially, it's going to pump what?
3 sodium in for 1
It's going to
calcium
allow 3 sodium to come inside the cell, down sodium's electrical and chemical gradient, right? So down its gradient. I guess actually at this point, if the cell is fully depolarized, it would be not against its electrical gradient, but it would still be against its concentration gradient. Either way, sodium's going to want to, if we could anthropomorphize, there's that word again there, Hunter. I always feel like I'm saying it wrong after I say it right. I mentioned this in class yesterday, Hunter. But it's going to allow 3 sodium to move down its gradient, down its concentration gradient from the outside of the cell into the cell, and it's going to use that energy to pump 1 calcium out. Right, so that's another mechanism. You also have calcium ATPases on the cell membrane that are literally just going to burn ATP to pump calcium out. And remember with calcium, okay, and this is assuming that you've already gone over the material. Remember that we have different mechanisms to decrease what we call, where we've been saying calcium, but it's free calcium or unbound calcium, because that is the calcium that's going to contribute to the overall calcium gradient. So you have proteins, which are called calcium binding proteins, in your SR that are going to lower the calcium. You even have some in your cytoplasm. And their whole point is to bind free calcium to lower free calcium so that it's not binding to troponin or it's not binding to calmodulin, right, in a smooth muscle. And that's another mechanism by which we can lower calcium levels. Some of these calcium binding proteins are CBP, literally called calcium binding protein, calreticulin, calsequestrin, some of these other molecules. There's other ones out there, but those are the main ones, okay? So now we've resequestered calcium and it's ready to go for the next action potential, right? So I think we've come full circle. Dom, I have not been, I've been freestyling, yo. So freestyling, that's how I roll. So can you tell me if we've missed any of the highlights between skeletal muscle and...
We have not missed any of the highlights,
Okay.
but the last couple things we want to talk about are the length-tension relationship with the relation to the sarcomere, and then isometric and isotonic contraction.
Very good. So once again, we're going to briefly go over this. Dom, do you feel comfortable talking about the length-tension relationship and muscle?
So, as we're talking about length-tension relations, sorry?
It's good over here.
Sorry, Hunter.
There you go. Give him some space. He's getting ready to work his magic.
So, we always want to talk about there's an optimal length for contraction. So, like when it comes to skeletal muscle, the greater we have a stretch of a muscle, the more optimal length, the greater contraction that we can have.
Are you saying optical or optimal?
Optimal.
Oh, I thought you
Optimal,
said
right.
optical. And I was
Yeah,
like,
no, it's definitely not optical. We're not talking about the eye here, right?
Man.
Right. Yeah, and so just to add on to that, skeletal muscle, when it's fully stretched, right? I cannot, unless I break my arm and take the muscle out or, really do some damage, I cannot stretch or cause my biceps femori to have any more stretch than it already has. And what Dom is saying is that at this point, at this maximal stretched point, right, my biceps, the actin and myosin overlap, right, they are at an optimal overlap for maximal force generation. Okay, that's what we're saying when we talk about optimal overlap versus not optimal. Meaning that if I have my biceps right here, say it's half contracted right here, and then I want to contract it all the way, I can't generate as much force, total force, going from here to here as I can going from maximal stretch all the way up to here. Does that make sense? So I just wanted to add that on there for that. Go ahead, continue on.
Yeah, so
Dom.
that's really it for skeletal muscle.
Right.
There is a principle when it gets to cardiac physiology, which you'll get into more into 641 about the optimal overlap, but technically the heart at its resting state is at a suboptimal overlap.
Very good.
So that it can stretch as blood fills the heart to a more optimal overlap. But you'll get in more to that
If
in
you're
641.
exercising, yeah.
But yeah.
Very good. Okay, and so that's the sort of the in crux, sort of what we're talking about with the length tension relationship. Remember, skeletal muscle is at a optimal overlap when you have it at its maximal stretch. When we talk about smooth muscle, because we talked about cardiac's at a suboptimal overlap whenever you're undergoing just resting state diastole. And that's good because if you want more blood to come in, it's going to stretch it to a more optimal overlap, which means you're going to get more force generation. Because you've got more blood, you need more force generation to pump that blood out, right? And so that's important to have with cardiac. With smooth muscle, remember that smooth muscle is kind of the outsider. It's the black sheep of the family, right? It is going to be able to, it has multiple lengths at which it can have maximal, or it has more variability in its length tension relationship, and it can generate maximal force over various lengths, right? So
Because it doesn't have a sarcomere.
It doesn't have a sarcomere.
So there are really not much limits.
That's right. And so because it doesn't have a sarcomere and it's not bound within the Z disc to Z disc space, right, or span, it actually has what are called dense bodies in there. It can actually decrease its length to a much greater percentage than skeletal muscle. So if this is 100%, if this is relaxed skeletal can maybe do this, maybe like 60%, right? It can go to 60% of what it would normally do. Smooth muscle, if this is relaxed smooth muscle can really contract to a much greater degree and sort of squish itself and shorten itself to a much greater degree than can skeletal muscle. Okay, and cardiac as well, because both of those are bound by the sarcomere, and the sarcomere is bound. The sarcomere unit is Z disc to Z disc.
I was gonna say the last thing we talk about, a very brief like I did in the microvid, a very brief explanation just to go over isotonic versus isometric.
Isotonic and isometric.
The way I described it in the microvid is isotonic. That means that the metric, the actual measurement, that's the thing that's changing. So isotonic, you're changing the length of the muscle. So that's like if you're trying to lift something or curl something, you're changing the muscle length. Whereas isometric, the length of the muscle is staying the same, but you're trying to apply more force. So like if you're pushing against a wall, or if you're like holding something, not stretching your muscles, but like you're holding a large amount of weight, if you're not changing the actual length of your muscle.
So you're contracting. You're contracting it, but with an isometric. So you're generating tension, you're generating tone, and it is contraction, but there's no muscle length. And the one thing I wanted to say is that anytime you have a contraction that is moving a load, right? You are going to use both isotonic and isometric. First, it's going to be isometric until you generate that force needed to move the load, and it's going to be, once again, isometric. Iso means same, metric means length, right? So your muscles are not changing in length with an isometric, but they are contracting. It's like if you're lifting, you want to do a I don't know what's a curl, right? You're doing a curl of 50 pounds. You have to go from 0 pounds to 20 pounds, to 30 pounds, to 40 pounds, to 45 pounds, to 47 pounds. You still have not moved that load, and you've not changed muscle length. Your muscles are contracted, and then once you get to right over 50, then it changes from an isometric to an isotonic because you're shortening the muscle length. Okay.
Again,
go ahead.
There's an easier way for me to think about that, and it's not really like just something to throw out. Like if you're moving joints, it's isotonic.
Not drugs.
No. I mean, like if you're moving the joints here, whatever, it's isotonic. I know. Right. It's isotonic. If you're basically doing planks, you're still generating a lot of force, okay, but you're not moving any joints. That's isometric. Does that make sense? This is how I think about it. It's a lot easier for me to understand it this way. Okay.
So if you're moving if your joint is engaged,
yes. If your joints are
movement, it's isotonic.
Yes.
Because it's as Caleb said, it's not isometric, right? So if you have muscle movement, you're engaging joints, then it's isotonic, right? All right, so isotonic versus isometric. Hopefully that gave you a little bit of a window into. Once again, this is not to reteach you the material. It's to kind of just review it. So, Dom, what else have we missed anything?
No, that's everything we've got covered for today.
All right. Well, thank you all so much. This has been a little bit of a longer podcast, I think. Maybe not. Time flies when you're having fun. We covered a lot of material, neuronal and muscle. So don't forget to, and actually mean it this time, because I say this in the lectures, but there's no like and subscribe. Don't forget to like and subscribe. Hit that, ring that bell, hit that button, I guess. And I'll leave it as I leave every class, right? Stay cool, stay in school, and don't do drugs. We'll see you next time on Eye on Physiology. Good job, y'all. Good job. Way to go. Well done.
I'll stay down here and upload